working in the lab right now. But, it's bare bones because of social distancing. So we've got to keep those limits going for a while, once hopefully the pandemic will blow over. So we're bringing you yet another groundbreaking journal article with the theme of aging and anti-aging, of course. And I will bring up the paper right now. Let's fiddle with my share screen. There we go. So it is from Stem Cell Reports. In vivo reprogramming ameliorates aging features in dentate gyrus cells and improves memory in mice. So this is Rodriguez Matayan, I believe is the pronunciation. Brought to you from the Serrano Lab and Jesus Avila. And the Serrano Lab, a little kind of claim to fame here is, I believe they're the first lab to have expressed Yamanaka factors, inducible Yamanaka factors under control of a promoter in transgenic animals, so mice, so in vivo. So, so that was an earlier paper, I believe it was a Nature paper, and here they're basically looking at the effect of expressing, inducibly expressing, stop sharing here, inducibly expressing Yamanaka factors in mice, and then looking at, well, what happens specifically in a particular region of the brain. And the region they're looking at is the hippocampal region, and I believe they're looking at the dentate gyrus and specifically also the subgranular zone. So this is where neurogenesis tends to occur, and also that region of the brain is responsible for memory formation. So clearly memories, the formation of new memories decreases over time as organisms age and other things happen. So this lab, they wanted to see what happens in this region of the brain if they induce Yamanaka factors. And just a quick recap of the Yamanaka factors. There's 4 of them. They were discovered by Yamanaka and et al. And Yamanaka et al. But the Yamanaka laboratory, and for which he won the Nobel Prize. And this is basically protein factors that can reprogram cells, reprogram adult cells into induced pluripotent stem cells. So a big deal. And it turns out that this aspect of reprogramming cells can potentially rejuvenate them as well. So let's take a look at the paper. So share screen right here. And if you have any questions, do feel free to chime in. So they look at a number of different things. So one thing to kind of keep in mind here is that when I was reading this paper, one thing that's been they're doing is expressing these factors throughout the mice. So this is not specifically, to the best of my knowledge, I read the materials and methods and pretty sure that's correct. They're not specifically inducing them under a promoter that's neuronal specific, right? So they're still being expressed throughout mice, through most tissues, and they do an RNA analysis to show that these factors are being expressed within neurons. But they're basically focusing on what happens with respect potentially to neurogenesis and also to neuronal migration, and also through modulation of epigenetic changes in the mice, and also do memories improve in mice as well. So they do a memory test as well. But just to recap, these factors are, I believe, being expressed in every cell. Okay, so first a little bit of background. So it's a pretty short paper, so it'll probably be done way earlier than usual. So figure 1, they're basically looking at 3-month-old mice and 10-month-old mice, so youngish mice and older mice. And this is just kind of to establish a baseline. So these are not Yamanaka treated mice, but they're basically looking at the dentate gyrus, this area in the hippocampal region, and they're looking at, they're doing immunohistochemistry here, staining for this marker of epigenetic changes of that. Basically, it's a methylation marker at histone 3, lysine 9, so triple methylation, and this goes down with aging normally. And they show later in the paper that they can reverse this with Yamanaka factor induction. So this basically correlates to, so a decrease of this correlates to well, increased transcription decondensation of chromatin, I believe. So, so they have this, they show here that in their mice, you could see you have a decrease and they're staining. So that you have this correlation with this change that's been observed in other cells as well, taking place in neurons within the dentate gyrus. So, so now to Yamanaka factor induction. So one thing I have to mention is they do let me see if I can pull up, I'm going to stop sharing, pull up the supplemental, because there's a modest supplemental section here. And let me see where it is. Okay, I don't know where the supplemental is. Anyway, but you're gonna have to take the work. But one thing to keep in mind is they do two types of induction here. They do continuous, meaning that the way that the Yamanaka factors are expressed is they put this antibiotic doxycycline into the water supply of the mice, and that switches on a promoter. So basically switches on a transcription factor, which then activates a promoter, and it turns on these Yamanaka factors. The other way is to basically sporadically, periodically, every few days, give them doxycycline and then deprive them of doxycycline for a number of days. And they do this at, I believe, at the beginning of 6 months, and then they continue that until the mice are 10 months old. The difference is that it's a very tricky protocol. If they give them continuous doxycycline treatment and continual induction of Yamanaka factors, the mice pretty much, I think mostly all drop dead within 2 weeks. So they die pretty quickly. So you can't really have continual expression of Yamanaka factors. So so this continuous was done. This was done for 12 days on 6-old mice, and then they quickly sacrificed the mice and then analyzed them, right? So the mice, really, most of the mice were kind of dying at this point. So when they do so they wanted to repeat these experiments later. For obviously, if you're going to be, using this as a potential therapeutic, right? So so you want to optimize your protocol so induction of Yamanaka factors doesn't cause the dramatic side effect of death. So you want to basically pulse it, and that's what they figured out a protocol later where they can induce this in mice and will have them survive. So anyway, let's take a look at what they see when they actually do a continual induction of Yamanaka factors and looking at immunohistochemistry in the dentate gyrus. So they look at various markers for adult hippocampal neurogenesis. So it's known that in this region, neurons can divide, and they wanted to see if the Yamanaka factors can actually promote neurogenesis. The bottom line is potentially no. So that's kind of the bad news. They're expecting neurogenesis, and these are various markers. So BLBP, that's an early neurogenesis marker. Whoops. They look at double cortin. They also look to see if, interestingly, for I don't know why the mice are dying, but it doesn't appear to be through apoptosis because there's caspase 3 activation doesn't go up, and this is figure 2C. P histone 3, which is also a marker, potential marker of, I believe, of neurogenesis. They have 2 different phenotypes, and I'm not entirely 100% sure. They mention it in the paper. They have a condensation phenotype and what they refer to as a transcription phenotype. So if you look at basically the immunohistochemistry, one is basically darker left than on the right. And that's just more staining for phosphorylated histone 3, which may be a later signifier of mitosis, they claim in the paper, and transcription phenotype, they mentioned that it might be the cells are still, I believe, in interphase and are not quite mitotic yet. So they might just be, they might be transitioning. So they don't have any evidence, any strong evidence that the cells are dividing. But the interesting thing is that when they look at some cells that are stained within the hippocampal region, and they do what's called a migration assay. So double cortin, so figure D and E, double cortin are stains that it's a factor within these neurons, so a stain, and then calretitin, which is basically another factor which is responsible for calcium modulation within the cells. They do notice that the cells have migrated, and the way that they quantify this is that when they measure, they have a little box here, when the cells are out of their region and are stained for these various factors, they score those cells as having migrated. So I'm going to stop sharing here. So what does that mean? Well, maybe the cells aren't dividing, but they have moved from one region of the hippocampus to the other. That's some evidence for neuronal plasticity. Certainly in memory formation and consolidation, neuronal plasticity is something that neurobiologists talk about, and that's the ways neurons basically make new connections with other neurons. There could be follow-up experiments to this, because I know they do this migration assay in this paper, and it's rather static. You see these cells that are normally in one region, and then when you after treatment, they're found stained in an adjacent region, and the numbers go up after induction of Yamanaka factors. I'm already thinking about other follow-up assays to neuronal plasticity. I think with increased neuronal plasticity, you have more formation of what's known as dendritic spines, which is basically more neuronal connections. So neurons establish more connections with other neurons. So it would be interesting to see a follow-up as to why the cells are migrating from that one region to the other, and if that's if as a consequence of that, more synapses have formed. So you can certainly stain for synapses and see if you have an increase in synapse formation. So that's that would be my comment there. So let's go back to let's go back to share screen, back to the paper. And as I mentioned before, if we look at figure 2F, there's a modest increase. Let's increase this here. It looks about, 1.5, almost twofold increase of the histone methylation marker H3K9, so lysine 9 triple methylation when you induce Yamanaka factors, suggesting that you have basically a reversal of this fact that happens with aging cells. So what that exactly means at the physiologic level of the cells, I'm not quite sure. They haven't dug down that deep. Let me pull up the chat here. Frederick Roslan at Facebook asks, could this reprogramming possibly be used as a treatment for depression also via increased neurogenesis? Don't know, because we haven't really, this paper hasn't established increased neurogenesis. So I don't know. We'd have, one thing I'd like to mention is there's other, so they look at various markers here of, it's like BLPB and other markers of, potential markers of cells that are transitioning to neurogenesis. One thing that, I would pose this as a, as a follow-up experiment is maybe something a little bit more generic to take a look at if there's any neurogenesis happening in any cells, could be glial cells, could be any cells within that region, using maybe BRDU or something like that to see if there's any actively dividing cells. That might be a follow-up. I'm not going to completely discount neurogenesis, just to state that the cells they looked at here with the neurogenic markers came up negative for neurogenesis, but that doesn't mean that it's not happening somewhere. It's not happening with other cells within the hippocampal region. Let's see. Follow up to this is possibly in conjunction with therapy steered towards new non-ruminating synapse formation. I'm not sure. I don't know what non-ruminating synapse formation is. I knew what synapse formation is. I knew what new synapse formation is. I'm not sure what non-ruminating synapse formation is. Do you guys know what non-ruminating synapse formation is? I'm ignorant of that. I don't know.
It's a new one for me, that.
Okay.
I'm just passing it on what's going on in the chat.
I know what a ruminant is.
Isn't it a type of cow?
It's a yes. It pertains to, I believe,
Cattle anyway, isn't it?
Multiple stomachs, yeah, animals that basically can redigest their food. But I don't think that's got anything to do with synapse formation. But no. Yeah, so it hasn't been definitively defined. So in answer to Frederick Roslin's question, it hasn't been definitively defined in this paper that that there's any neurogenesis occurring. Potential neuronal migration, yes, and a reversal of the decrease of the triple methylation at histone 3 lysine 9, but that doesn't mean that neurogenesis isn't occurring. Like I said, I'd be interested to see maybe using a BRDU assay, to see if there's other neurons that might be dividing. Okay, let's go back here. So that's continual induction. So 12 days on a 6-month-old mice. So what else did they do? So now they do cyclic induction. So same thing, but now cyclic. So now the mice are not being outright killed by the therapy or the treatment. So this is 3 days doxycycline administration followed by 4 days of doxycycline withdrawal during a 15-week cycled protocol, which is good because the other mice would be quite dead well before 15 weeks have elapsed. And I believe what they do here is they look at older mice as well. These mice here were 6-month-old mice, and this protocol was initiated on 6-month-old mice. So the mice were, when they analyzed them, I believe they were already 10 months old at this point, right? 15 weeks. So they were much older. The bottom line is, again, they don't see any markers that suggest that there's neurogenesis taking place here. So double cortin, BLPB, so early markers of neurogenesis. But they do see, again, an example of increased migration of cells within the dentate gyrase when they basically stain them and measure them and show that they've moved, I believe, like 30 microns from one area to another. They do see an increase in methylation, triple methylation at histone 3 lysine 9. So basically a repeat of what they've seen here, except for, the dramatic effect of this being now potentially therapeutic since it's not killing the mice. Okay, so kind of the most interesting thing is, papers always put the most interesting thing, leave the best for the last, which is, all right, you've got these changes happening. How do the mice actually perform? And they do a memory test. So they do this memory index where they have it in the protocol. There's a little picture of a mouse in a pretty bleak box, I guess, up there in the upper left-hand corner. And they put 2 objects in the mice in the box, and one object the mouse has already seen before. The other one is a new object. And then they measure the amount of time that the mouse is basically sniffing the new object, and they repeat that. And the amount of time that the mouse goes to a new object is indicative of memory formation, right? So if they do this, if they move the mouse out of the box and they put in a new object and the mouse back in, a 2 hours difference between wild type and Yamanaka factor induced, there's really no difference in memory formation between these mice. However, 5 days, you do notice a difference. So there's, indication that you have long-term memory formation in these mice. And interestingly, this plot here that they have in C, figure C, they do these assays and they have the memory index based on how much time the mice sniffed around on the new object. And they show a correlation, they show somewhat of a correlation with messenger RNA levels that are basically indicating levels of, because you have, different transgenic mice in this study. So each dot is a different transgenic mouse, and you have different, kind of fluctuations in expression of Yamanaka factors. And it appears to be, somewhat of a correlation there with an increase in Yamanaka factor expression and performance on this memory index test. The last thing they do is they look at, so one thing that's noticed also, and that one thing that's required for memory formation is, I believe there's a class of neurons, I was gonna say GABA, but it's not, is it GABAergic neurons? I'm mixing it up, geez, where is it? So what's that subunit of? Glue N2B, it's not GABAergic, it's, NMDA receptor. Yeah, sorry. So it's the NMDA receptor, and that typically goes down when organisms age. And upon induction of Yamanaka factors, this NMDA receptor, which is actually important for memory formation, goes up. So they speculate that maybe it's got something to do with the NMDA receptor. Of course, a good follow-up experiment is to inhibit this receptor. Maybe, I would throw that out there with an antagonist and see if this is blocking memory formation, if it brings it back to wild type. So the bottom line is Yamanaka factor induction, both long-term induction and also pulsed induction, seems to improve memory formation in mice. You do have a reversal of the effects that you see at histone 3, triple methylated lysine 9. I'm not sure how that might correlate. So there's a lot of questions that still need to be answered. I think it's a very good first step forward to figuring out if reprogramming of neurons can have the same beneficial effect as is seen in other tissues when cells have been reprogrammed using a variety of factors such as Yamanaka factors in aging. I would be also intrigued to see, this, what the effects would be if you specifically express these factors in a neuronal only fashion and not throughout the mouse. So these factors are being expressed in neurons, but the caveat here I would say is that they're also being expressed throughout the mouse as well. So it's hard for me to tease out what other effects from other distant organs there might be. I'm assuming these factors are also being expressed in the vasculature, right? That and so on and so forth. So so that's really it. Is there any questions? We have one good question there related to depression, but I guess the answer is that we don't believe that neurogenesis, at least according to this paper, is being affected, although they only looked at certain neurons. It could be neurogenesis happening elsewhere. They could use BRDU staining to maybe see if there's other neurons, maybe glial cells that are being affected by the Yamanaka factor therapy. Any comments? Like I said, this was a short paper. They had the transgenic mice, they had the technology, and they wanted to see what happens. And there is some tantalizing evidence here that we may have, well, certainly memory improvement in these mice. So that's a positive. But how exactly this memory improvement is happening, I think it's still there's still some questions that need to be answered, and a lot of good experiments that could be designed to answer those questions.